Thank you, Elliot, and thank you all for the opportunity to present today. I too will not have slides, so you can continue to take your nap if you need. As an epidemiologist who has conducted numerous real-world evidence studies to understand the burden of disease or to study the effectiveness or safety of a therapeutic intervention or to evaluate the quality of an intervention for patients and other stakeholders, I think I can say I have a fairly solid understanding of what constitutes high-quality real-world evidence. But I can also say that motivating my colleagues and fellow leaders within an organization to embrace and endorse real-world evidence-based approaches, particularly within the setting of a regulatory engagement, is really not that easy. And it is my impression that biopharma is not the only environment where these challenges exist. I would proffer that there are a number of existing obstacles that need to be overcome for real-world evidence-based approaches to gain traction and to ultimately be more widely accepted and endorsed. First, I think there's a general lack of understanding of non-interventional research methodology. There are specific communities, such as the epidemiology and health services communities, who are well-trained in these approaches, but they represent a minority of folks working in the clinical research area. Many, if not most individuals working within clinical research are unable to distinguish between higher and lower quality real-world evidence. As a result, many of the team members ask, what is a reliable or trustworthy approach, which often can derail conversations. I think there's a general mistrust for data not collected in the highly protocolized and monitored environment of a clinical trial. So oftentimes the questions, are the underlying data good enough? And what is missing are asked. And fourth, today there is no clear regulatory pathway for real-world evidence-based approaches. And in the absence of such a pathway, there is really little willingness to embrace and mobilize resources against an approach that does not have a reasonable probability of success. So with these challenges in mind, I'd like to offer some thoughts about the current state of pharmacoepidemiologic research methodology, to recognize some current examples of high-quality real-world evidence, to highlight the importance of data quality and offer support for continued efforts to harmonize making interoperable data systems across the healthcare environment, and to offer some ideas for advancing the understanding of real-world evidence, all with the goal of getting us unstuck. So over the past 3 decades, the application of pharmacoepidemiologic research methods has improved substantially. General design principles are more broadly understood, are more routinely implemented, and are part of the curriculum in most good training programs. For example, studies that employ a new user design, that select an active comparator, and compare against the same modality, address many of the now recognized biases that may have led to spurious results in the past. There's a greater understanding of the sources of bias and approaches to bias adjustment, whether that be propensity scores, high-dimensional propensity scores, or inverse probability of weighting. These are now more commonly applied in our research. And I would say with the availability of newer techniques, such as quantitative bias analysis, we have the ability to empirically evaluate the impact of hypothesized or real sources of bias, such as an unmeasured confounder or outcome misclassification. So today there's greater understanding of these key design and analysis principles and how to use them to answer drug effectiveness and safety questions. These principles are expertly communicated in currently available textbooks, such as the third edition of Modern Epidemiology, and in guidance documents such as GRACE and STROBE. And so the information is there. Second, it may be important to distinguish between what constitutes higher versus lower quality real-world evidence, and whether that evidence is designed to address a safety question or a question of effectiveness. For more than 2 decades now, drug safety questions have often been addressed using large healthcare databases that employ rigorous pharmacoepidemiologic research designs. The list of safety questions where rigorously conducted real-world evidence-based studies have produced high-quality evidence. One might say evidence that's consistent with clinical trials designed to answer the same question. They continue to grow. These same research methods are those that have been adopted in the Sentinel system. And for that reason, we may have, within the safety domain, a good understanding of what might constitute high-quality real-world evidence. However, in the context of effectiveness, the situation is potentially more nuanced. Obtaining marketing authorization for a new medicine or expanding an indication leveraging real-world evidence-based approaches may be limited to very specific scenarios. As outlined in the Sherman article in the New England Journal of Medicine, these may be limited to patients who have significant unmet need, few available treatment options, and a very poor prognosis, such as rare diseases or uncommon tumor types. A recent example is the approval of Blincyto for the treatment of relapsed refractory Philadelphia chromosome negative acute lymphoblastic leukemia. Here, a historical comparator group was selected as a control arm to a single-arm phase 1b, phase 2 study. The large treatment effect observed in the Blincyto arm relative to that which was observed in the propensity score matched historical comparator was deemed of sufficient evidence and sufficient size to enable marketing authorization. What is fortunate here is that these results were corroborated by the soon-to-follow Tower study, a phase 3 2-to-1 randomized study examining the same treatment effect. So I think we have examples that if we rigorously evaluate and identify the key defining characteristics, we may be able to begin to quantify or qualify what is high-quality real-world evidence. Third, promoting data quality standards and the interoperability of systems across the various settings of care is fundamental to the advancement of real-world evidence. The richness of data captured in electronic health records, information on disease severity and progression, on laboratory markers, on diagnostic tests, on genetics, coupled with information through wearable devices, and with the incredible advances in technology such as deep learning and natural language processing, forecasts an emerging information ecosystem that can contribute to significant advances in our understanding of the effectiveness and safety of medicines. But for this to occur, data standards that meet regulatory requirements will need to be adopted across the various settings of care where RWD are collected. These systems will need to be able to communicate with one another or rely on technologies that can aggregate real-world data from multiple settings of care. But with the adoption of these data standards across the various settings, we can begin to build greater trust in the accuracy and reliability of real-world data and the evidence derived from it. Lastly, considering the various challenges that the adoption of real-world evidence in regulatory decision-making faces, it seems prudent to advocate for an educational campaign around real-world evidence. Can we build a curriculum championed by the DIA or some other body that can provide our colleagues in biopharma, in regulatory agencies, contract research organizations, and other groups with a greater understanding of what constitutes real-world evidence, whether it be pragmatic clinical trials or pharmacoepidemiologic research designs? And as part of that, is there an opportunity to convene workshops where these same groups of folks meet to discuss the merits of different study designs to address a given safety or effectiveness question? Drawing on the significant expertise that lies within the halls of our respective institutions, we may be able to elevate our common understanding and trust in real-world evidence and advance our ability to bring safe and effective medicines to patients using this type of evidence. Thank you for the opportunity to present.